So it's my privilege to give our regular update on behalf of the Division of Extramural Research and Training. And let's see if I can go to my next slide. There we go. I've got a number of things to cover today, so I'm going to move through this pretty quickly. And the first thing I'd like to cover is going over some staffing changes that we've had in DERT and let you know some goings and then a couple comings as well. So the first is members of the staff who've left us since the September council meeting. And the first is two retirements that we had. Pat Maston, I notified you about in September of his intention to retire at the end of the calendar year. So Pat joined NAEHS back in 1998 and for the last 10 years had been working as the Deputy Division Director for DERT. Our other retiree was Bill Sook, who many of you know for his role as the director of the Superfund Research Program, was hired by NAEHS in 1987 and is the only director that the Superfund program has had in its history up until now. So we wish both of them, the best of luck as they move into a new phase. I know both of them have lots of plans. Pat's been spending lots of time in his studio painting and playing guitar, and Bill is being Bill and doing all kinds of environmental health things. We've had several other staff members who've left us for other personal and professional opportunities that I wanted to update you on. So the first is Helena Kennedy, who worked in our program analysis branch since 2018. She accepted a job with the TSA, the Transportation Security Administration, managing multi-hundred million dollar procurements for them. So she started in January of this year. And Tina Powell, who many of you may not have interacted with very much, but Gwen and I worked with her very closely. She's been working in support of the division director for several years. And she left us in November for a variety of health and personal reasons. Demia Wright, who worked in our worker training branch. She joined us in 2015, working as a public health educator. And she went back to CDC, to her roots, and also in November. And then Sarah Amalegby, who has worked with us for many, many years, actually was trained in DIR. And worked in support of several of our programs as a contractor and officially joined DERT as a federal employee in 2020 in the Hazardous Substances Research Branch, working on the Superfund Research Program. And she took a position in the Office of Strategic Coordination, which many of you may know by a different name, the Common Fund, working as an evaluator there. And then two staff, one new staff and one person in a new role. So Gary Johnson, we're glad to have on board working in the worker training branch, working on the worker training program, where he will be working as an industrial hygienist, working as a program officer for both the U45 and the UH4 grants within worker training. He comes to us from Customs and Border Protection, where he worked as an industrial hygienist, core and manager of the National Medical Surveillance Program. And prior to that, he worked in the Department of Labor at OSHA. And then the other, Rick announced a little bit this morning, Michelle Heacock actually will on Sunday, this coming Sunday, will assume a role as the acting chief of the Hazardous and Substances Research Branch and director of the Superfund Research Program. So we thank Michelle for stepping up. She's already, we started transitioning her into some of the division leadership activities over the last couple of weeks since we made the selection. And Michelle will be continuing to manage her portfolio. So the Superfund grantees that had Michelle as her program officer will continue to work with her, and she'll also continue on several of her other duties for the division. So my next theme, you may pause here for a minute. This is one of my statutory requirements. Every year I have to bring to you the council delegated authorities for your review and approval. So just as a reminder, these council delegated authorities are responsibilities that normally reside with council that in the name of efficiency, we ask that you delegate those authorities to us. And there's 5 things that we ask you to allow us to do. The first is for the change of a grant from one grantee institution to another. The second is a permanent change of the principal investigator of a grant. The third is a temporary change of the principal investigator. This is typically either in the case of a sabbatical or an extended medical leave. And then the fourth is a no-cost extension of a grant into an additional budget period. And then finally, the extension of a grant with additional funding. So we have not proposed any changes to the council delegated staff actions for this coming year. We do have one change into the document, however, that I want to highlight to you. And this is under the section for special counsel review policies for applications from investigators with more than now $2 million total costs of existing NIH funding. This is a change from the previous standard of I believe it was $1.5 million of direct costs to the $2 million total cost. And this change is made to comply with guidance that we are receiving from NIH on the special counsel review policy. So with that, I will call for any discussion on the council delegated authorities from council. And I've asked Cindy Lawler, since I can't see the gallery, to tell me if there are any hands raised for comments, questions. Not seeing any. If there's no council discussion, may I have a motion to approve?
So moved.
Thank you, Terry. And a second? Gary, thank you very much. And if council members would please vote in the electronic council book. Okay, thank you. We have quorum on that. So my second requirement in the February council is to bring you a report on our 22 previous fiscal year funding, a budget wrap up, if you will. So this slide gives you the 30,000 foot overview. So in 2022, we received just under 1,300 applications for new grant awards for the division. Of those, we funded a total of 250 competing awards, most of which are in the research project grant category. And I'll tell you a little bit more about that in a little bit. But we also funded 29 training grants. That's both institutional and individual awards. 34 awards in the other research category. That's primarily career awards, 6 center grants, 26 small business grants. Our average competing cost for those awards was $450,000, which is slightly lower than our non-competing out-year commitments of $477,000. For the year, we were able to maintain our longstanding fixed pay line at 10th percentile for investigator-initiated R01, R03, and R21 awards. And that resulted in as overall success rate NAEHS for R01s of just over 15%, and for RPGs just over 16%. Those numbers are slightly lower than the NIH average. Looking at the distribution of grants, so this the previous table as well does not include any funds that are going into the Superfund program or the worker training program or our TAPs that we have to pay up to NIH for management. So our total extramural investment in 22 is just under $395 million. Of that, 71% is in the research project grant range. 10% is in our centers program, 6% in training, 5%, which is a statutory level that's set by Congress, is in our small business awards. And our other research support is 8%. And that's a number that has been steadily growing over the last few years as we have been emphasizing really research capacity building projects. So looking specifically at our research project grants and the distribution of awards within that 71% of our overall budget that's in RPGs. The overwhelming majority of that is in our R01 line. That's your standard research project grant. The second largest bin of that is our R35. Those are the river awards, which we began about 6 years ago now. And that has steadily grown to be almost 10% of our research project grant line. And now actually exceeds the amount of money we're putting into small grants like the R21 program. Our UO1 cooperative agreement mechanisms is next, and then we lump all of the other research project grants, which is mostly things like conference grants, R56s, R03s, and the area awards, the R15s, into that remaining $60 million. So here we're looking at that same overarching $280 million, 71% of our grants budget, just to highlight that actually most of that, 208 of the $280 million is our out year commitments for previously funded R01 for the most part awards. So 436 of the total 590 awards made, and only 160 of that is made into competing awards for a total of $72 million. Those numbers are fairly flat over the last several years, with accounting for noise. So in 2020, our average cost of award was actually slightly lower. But our competing award budget was actually about the same as it was last year. In 2021, our available budget for competing awards was slightly lower due to some higher out-year commitments, but our average cost of award was higher. Therefore, the total number of awards that competing awards that we issued in 21 was slightly lower than last year. And so we saw a bump up in our budget, and we were able to make a higher number of larger awards last year. So I want to, excuse me, call attention to our solicitations. So we do a number of different kinds of RFA based solicitations in the division. Many of these are joining institute programs that are led by other institutes. So we will make a very small investment of those 1 or 2 awards here and there. Some of these are long-standing so-called permanent programs that we invest in, things like our time-sensitive awards, the ONES program, the River program, where we will have continual recompetition of those programs. And then a couple of these are one-time solicitations, things like the Inflammation Resolution Program, RESTORE, or the Functional Genomics program that we did last year. And those you will typically see a larger number of awards and a larger financial investment in any given year. And just to highlight in the corner, it's a small font, but the number of awards and the total investment in our solicited program, again, has been fairly consistent over the last several years. So here I wanted to highlight, I commented that we maintained our fixed 10% pay line for several years, and that does not spend all of our budget available, traditionally does not spend all of our budget available for competing awards. And so I just wanted to have some transparency in how many of those projects are within the pay line, the yellow lines here, or are what we call raise to pay or select pay projects, where we kind of make an assumption that, in the 10 to 20th percentile, the quality of the projects are good, and we should be making selections for things that align with institute priorities or some subtleties of the review and the quality and gaps that those projects are filling within our portfolio. So it's, as I said, 71% of our budget is in RPGs, which means 29% of our budget is not. And so the largest single bin of that is our centers program, where we have a total of 24 P30s that we fund for just under $40 million investment. Our training grants are both individual and institutional awards, which comes in at $23 million. Our small business, which comes in about $21 million. Our other, which is mostly career awards, but as I said earlier, that is a line that has been increasing as we have been doing things like the HERE program, our cohort maintenance programs, our R25 short-term training courses. So these are our projects that are really focused on supporting the research infrastructure and building that in the future. And then to highlight the Superfund appropriations. Again, we support two programs with our Superfund appropriations, the worker training program with a total budget of $27 million, and the Superfund research program with a total investment of $51 million. Again, the majority of the worker training program is in the U45 program with a small amount of SBIR that they support through their e-learning program. And the Superfund program is mostly in the P42. It also does support a small R01 program, a small business program, a short-term training program, and then occasional R21s and R13s. These programs are almost entirely solicited research for the Institute. So I also wanted to highlight an effort that we started about 3 years ago that I hope you will appreciate, which was an effort to really speed up the timing of awards and being able to issue more of our awards earlier in the fiscal year so that applications are not sitting in a queue waiting to be approved. And so I wanted to bring this to show some of the success that we've had. So historically, from 2017 to 2020, we were by this time of year, by the end of January, paying somewhere in the 15% of the number of awards that we anticipate we're going to fund in the year. And recently, we've been trying to push more of our work into the first quarter of the year. And you can see from 21 to 22 to 23, we're now funding almost 30% of our anticipated number of awards through the year. So we're really starting to see some benefits of this, taking some of the workload off of staff, freeing up some capacity to do more work later, and also getting money out to you, the research community, faster in the year. So I want to look a little bit forward now into the things that are going on in fiscal year 23 with our funding opportunities coming up. Many of these are all on the streets right now. So we have some of the same players in our solicited research, our time-sensitive program, the core centers, the ones, the e-learning program, and our RISE program. We've also got our Victor competing this year, our cohort maintenance competing this year. River is coming to closed session today. We've also got the climate U24, which is coming to closed session today, and an SBIR program that is competing this year. And then Rick verbally alluded to this and was running a little short on time, so did not highlight. We have one significant program that we joined from another institute last year, which is a program led by the Human Genome Institute with us and NCI as partners, really looking at multi-omics for health and disease, a series of population of RFAs to really build capacity to do multi-omic integration. And we'll be bringing this to May Council, but I can tell you there was a very robust response from the environmental health sciences community to this program. So we're really looking forward to seeing the results of this. Then another thing I wanted to highlight to you was another results of efforts that we've been making, which is really trying to beef up our diversity supplement program. So we made a commitment 3 years ago to increase our investment substantially in our small business programs. And you can see over time, we've gone from a diversity supplement program that was just around $1 million to one that is approaching $4 million annually, with almost twice as many awards that we are issuing to support a professional pathway for to build the diversity within the environmental health sciences community. So another thing that I tend to bring we tend to bring every council round, just for your awareness, is highlighting some activities that the division has been doing recently and some things that are coming up down the pike. And as always, we've been very busy in DERT. We've done a number of workshops and holding a number of grantee meetings, highlighting just a couple. I think many of you are familiar with the efforts of the ELK, the Environmental Health Language Collaborative, which has actually been holding a series of discussions through the year. We also have had a meeting of our Driven, that is our diversity supplement trainees. We've been really working to form a network of them, so it's not just funding more awards, it's also
we received the agenda. It was sent out just
thank you. And then things that are coming up down the pike, we have a number of grantee meetings that are coming up in the coming months for our worker training program, our telomere grantee, which is a partnership with NIA that I think Dr. Hodes will speak about tomorrow. An annual meeting of our environmental epidemiology and autism risk meeting. That's actually going to be in Sweden. And then we're doing for the first time a joint meeting of our ones grantees, our cream of the crop early stage investigators with our river investigators, our People versus Projects program grantees meeting, hopefully in May or June. We haven't set the exact dates for that. So I'm wrapping up. I actually went through this faster than I was expecting to. So just a couple of things to highlight to draw your attention to some recent news. This is mostly coming from NIH and things that we could use your guidance on. So the first one I want to highlight is clinical trials. So a few years ago, NIH put increased emphasis on the definition of what is a randomized clinical trial. And recently, we have been running into a number of issues with applicants who are proposing to do what meets the definition of a randomized controlled trial in response to an RFA that does not allow clinical trials. When this happens, almost 100% of the time, that application will be rejected and returned without review. So this has become quite a pain point. Many intervention trials, so we're not testing therapeutics, but we're testing policy or behavioral interventions, do meet the definition of an RCT. And when you apply to a clinical trial not allowed FOA, NOFO, that application will almost always be returned by NIH. So I just want to draw attention to some resources that are out there from NIH, the clinical trial decision tool that'll help you know whether what you are doing meets the definition of a randomized clinical trial. I also want to highlight a couple of things that are coming down the pike for peer review. This peer review obviously is a topic that gets a lot of attention. And one of the things, the big things that is coming, probably the biggest thing that is coming, is NIH has recently released a request for information, a proposal to change the way NIH proposals, applications are reviewed. So we still have to follow the 5 scored criteria, That is significance, innovation, approach, investigator, and environment. Those are still part of the review consideration, but the new framework reworks those into 3 new factors. The first is called the importance of the research, which is an amalgamation of the significance and the innovation. You could think of this as, should you do this work? The second factor is the feasibility and rigor, which aligns with the approach from the previous criteria. And that's the, can you do this research? So those 2 factors will receive an independent score. And then the third factor, which is the expertise and resources, which is the former investigator and environment criteria, will either be rated as appropriate or additional resources needed. They will not receive a score. And again, the project will receive an overall priority score, much like today. But again, you'll only receive scores for those first 2 factors. Can you do it? Or should you do it? And can you do it? The score may also be impacted by some of the additional review criteria that we have now, things like human subjects. But most of the additional review considerations, things like foreign applications, the budget, animal concerns, those kinds of things, will no longer be part of the review discussion, and those responsibilities will be placed on NIH staff to do a review of those considerations. So we want your input on these new review criteria. As I said, there's an RFI that is out through March 10th for feedback on this new approach. There are some other modifications that are under consideration, such as review of clinical trials, and how things like the plans for enhancing diverse perspectives into the review criteria. So the URL for the notice is here, and please do follow the link, provide your feedback. I think this is a very positive change to peer review for NIH, but I think we're going to need feedback from the community to really make this new policy work for the field. The second thing I want to highlight is an effort that stems from the NAEHS. So this is the Trans NIH efforts on addressing diversity, equity, inclusion, and access. The Extramural Committee has really placed a significant priority recently on removing bias from peer review. And so there are 3 layers of the work that is going to be implemented as a result of the NIEHS efforts. And the first is bias training for both SROs as well as reviewers. And really trying to highlight biases is natural. We all have biases. We have to be aware of them, and we have to not take action on behalf of the biases that we have. The second is really strengthening our reporting mechanisms for bias in peer review, and focusing initially on CSR programs. To really build out a process by, if you are serving as a reviewer and you observe people acting on bias, that there's a way for you to report that into the peer review process. And then the third is promoting a culture shift in review panels by really emphasizing the value of diversity within the review orientation, and really helping us be more receptive to people who are less like we are, or other ideas in the review. And if you have questions on this, we have one of the experts in the room, Dr. Alfonso Latoni, who is the chief of our review branch, was on the committee that developed these recommendations. So he is here to answer any questions that you may have about this. So the last thing I want to highlight to you is some significant recent guide notices that I think I also hope that you will come and provide some additional input to us. So the first, for those of you conducting vertebrate animal or cephalopod research, NIH recently announced this is not a policy, but it's a recommendation. We're encouraging our grantee community to adopt the Arrive Essential 10 checklist in any publications that result from research supported by NIH using vertebrate animal and cephalopod. These 10 criteria that they're asking you to explicitly address in your publications are really intended to promote transparency and rigor and reproducibility in animal and cephalopod based research. So I've got the notice number here. Please go look at that, and I encourage you to adopt the Arrive Essential 10 in your publications. And then the final is a request for information that was just released, I believe, last week on re-envisioning the U.S. postdoc research, training, and career progression within the biomedical research enterprise. And in particular, we're really looking for feedback from graduate students, current postdocs, recent postdocs, in understanding the pressures that they are under, their needs in understanding career progression, things that we as NIH should be aware of as we're creating programs to support and foster them, and really helping us you work with the broader community to reinvigorate postdoctoral education in the United States. And with that, I am done with slides. I will say thank you for your attention and happy to take any questions. I actually got through that a lot faster than I was expecting to, so we've got a little bit of time. Cindy, do we have any hands up? Irva, use your microphone, please.
Yeah, I'm a little interested in the ONES program and what how the rate of success compares to, I don't know, say K-9-9s or just what is the success rate in some of those training, career development type of programs.
So are you asking about the success rate of getting a ONES application or of going from a ONES to a subsequent R01?
I guess both, actually. Yeah.
Yeah. So I don't know if Christy or Jenny Collins, or Carol, if you know those numbers off the top of your head. I think that the success rate for ONES applications historically has been pretty good, getting that initial R01 through the ONES program. And we did an evaluation of ONES probably 5 years ago. Christy, I see you're there. Christy, help me.
I'm not sure how much I can help off the top of my head. We have done an evaluation of those things, and we try to keep up with them and present them to the ONES community every year when we get them together for a grantee meeting. So, but we'll have to, I've written it down. We can get back to you with that information after council.
Yeah, I do, just kind of emphasizing, K-9 is a postdoc award. ONES is an independent investigator award. So they're not directly comparable. But that question of conversion from an R00 to an R01 or renewal of a ONES award. I do recall from the evaluation that the renewal rate for ONES investigators was very high. Almost all of our ONES investigators have received a subsequent R01 from NAEHS.
Well, I guess I was actually interested in sort of both that and among applications for ONES awards. I feel like I've known a lot of people who have tried, I didn't necessarily see everything what they submitted, but were people that I thought were pretty outstanding and tried maybe multiple times and didn't get it, so I was just curious if
Yeah.
that just happened. to be
Yeah,
the
and I think one of the questions
small
that we did
sample
look at in
number.
that one's evaluation was the success rate of people who received a ones versus the next,
Right,
5 people.
right.
And I don't recall the conclusion of that, so I don't want to speak
Yeah.
without looking back to the evaluation. Trevor?
I actually have two questions, a little bit unrelated. You talked about the new criteria that R01s might be reviewed under.
Mhm.
My understanding is that the concept of removing the expertise and resources segment to an unscored one was to unbias the review in terms of investigators that may have the very substantial track record versus those that do not. So how will that affect the biosketches going in for these applications that have been thought through?
Alfonso, do you know an answer for that question? Irva, can I get you to turn off your microphone? Irva. Thank you. It went right back on. There you go. Alfonso, do you know if I don't think that there were any changes proposed to the biosketch.
Not as of now, right? Because that's still in the workings,
Yeah.
right? And there's the RFI outside. But as of now, I have not seen anything on it changing anything in the biosketch, so I can't.
Because if the intent is what I said it is, then, it's natural curiosity that will let you look at the biosketch, and that is going to, perhaps bias your score when you don't want it to.
Yes. So Alfonso, maybe the other thing that you can comment is the anonymized review that I know that they've been doing through some of the common fund programs and conversation that RPC has been having about anonymized review.
Since we don't do it in our institute, and I think it's only done at the CSR. I don't really have nothing to add to what is. Are they still doing that? I'm not even sure that it's happening to an extent where the data are significant and can say something.
Okay, if I'm allowed, my
Yeah.
second topic was the RFI for postdocs. So
Yes.
I'm sure we could spend a whole day talking about postdoc issues.
Yes.
But one thing that does come to mind is, for example, at my University of Pennsylvania, we've just gone through a market adjustment in salaries for staff members, which now means that an incoming technician with their benefits, their total compensation is going to be very close to the year zero postdoc. And that obviously creates equity issues. And so one of the forces we have here are the stipend levels for NIH postdocs. And I know they're minimum and can be supplemented, but it kind of raises the issue of where those supplemental funds would come from as well. So I think as this conversation moves forward, some budget modeling is going to be required in terms of these stipends being increased as they have to.
Yeah, and I think that's exactly the kind of feedback that we're looking for in the RFI. I do think that there's, budget is a very complicated issue because it's, not set by us. I think, it's easy to say we need to bump up the salaries, but that money has to come from somewhere. So, yeah.
Hands up.
Gary.
Just following up on that, I think that, thinking about all the training grant director meetings that I've gone to and the trends we've seen over the last couple decades is I do think that the T32 directors are a good source of information on this because we've seen shifts in the postdoc stipend and things. I'm at a place that's unionized now where we're coming up with, $12,000 to $15,000 supplemental funding that can't come from the grants. And seeing this compression of the number of slots because of increased cost. And so like you said, it's a fixed size. And so if we say we have to, if the main complaint is, oh, we need to have the higher base salary, it's gonna mean fewer slots. And we need to figure out some way to, I'm very concerned with the sort of the supplementation part, because it's really getting to the point of what it does is it squeezes out junior faculty from taking people on training grants. And that's not what we want to do.
That's not what we're doing.
My other comment was about the R35s. Like there's probably not enough to influence it yet, but the thinking is that when you award the R35, you're pulling out these high performance investigators that tend to have a lot of RO1s.
Yeah.
Are you seeing it help the people that aren't the R35 recipients? Like when you look at the RO1s that they would have been competing for, is it freeing up slots for those people that are converting from K99s and things like that?
Yeah, so anecdotally, yes, I think we've seen some cases where, applications have still come in, but they've had a PI who was not the R35 PI. So it's creating an opportunity. We haven't done a formal evaluation of that just yet. We're coming up to we started that in 2018, I believe. So 2017, maybe. We're coming up to the phase where the first cohort of river investigators are going to be coming in with their renewals. And so, it's time for us to be looking at the program and really seeing where we're seeing those kinds of benefits and also the research impacts that we're getting out of the program. There are no other questions. I will wrap up and I will turn it over to Daryl to introduce our DIR speaker.
Thank you. So it's a pleasure to introduce today's DIR speaker, Shamal Padada. So Shamal received his PhD in 1983 from University of Pittsburgh, and he's currently a senior investigator in our biostatistics and computational biology branch at NAEHS. He recently relocated here from NICHD, where he was chief of their biostat and bioinformatics branch. And before that, he was chair of the Department of Biostatistics at University of Pittsburgh, his alma mater, which is one of the largest biostat programs in the US. And before that, he was here at NIEHS. So he's kind of come back to us recently. Shamal is well known for his pioneering methodologic work in analyzing microbiome data. Some of these methods developed by his group are among the standard methods for analyzing these complex data sets and have been integrated into a widely used software program called CHIME, which is developed by the Knight Lab at UCSD. Shamal has received numerous honors and awards, including being elected as a fellow in the American Statistical Association. And the title of his talk today is Gut Microbial Composition is Altered Prior to Seroconversion in HIV Patients. So, Shamal?
Thank you very much for your kind introduction. I'm really excited to present my recent work in this area of gut microbiome in the context of HIV infection. So human health is a function of a lot of different factors: physical activity, diet, chemical exposure, stress, circadian clock, and so on. And it's also a function of genetics. And for the last 15 to 20 years, people have started recognizing that our internal environment is just as important, the microbiome. And surely, because if we have about 10 trillion human cells, we have about 100 trillion microbial cells. And in terms of the genes, if you have 20,000 human genes, roughly speaking, then you have about 2 to 20 million microbial genes. And these numbers are updated all the time. So this is just a rough estimate. So clearly, the microbiome is an important component of our body, and that plays a major role in addition to all these factors. So in fact, our human health can be thought of as a complex interaction between our genetics, the external environment, and the internal environment over time. And in my research program, I've been developing methods in the past several years to answer some of the questions or the data that are generated through these various studies to get to this point. So today's talk is about microbiome data. Briefly, the microbiome data, what we observe are really relative abundances. So when you process any of these through these technologies, the observed counts that you get are counts which are relative abundances and not the true abundances. So everything should be thought in compositional sense. Things are relative to each other. As a consequence, you cannot use any of the standard statistical methods or the standard software like the t-test or NOR regression or any of those kind of things. And so we have been developing methods, as Dr. Zeldin just pointed out, over the past several years, how to analyze these data. And we were among the first few groups which actually identified that these data are compositional and therefore new strategies are needed to analyze these data. And today I'll not be talking about the statistical methods that we developed or used, but instead I'll be describing to you the results of the methods that we developed. The methods that I'm going to talk about today was recently published in Nature Communications with my student. So a few years ago, as Dr. Zeldin pointed out, I was at University of Pittsburgh, and I was having a conversation with Dr. Ronaldo, who is an expert in infectious disease, especially HIV. And we were discussing various opportunities to collaborate. And one of the questions we both were trying to understand is why some people, when exposed to an infection, develop the disease, whereas the others don't. Ignoring the factors, external factors, what is going on in the body that makes them different. So we wanted to understand that. So that's what led to this study. So Dr. Ronaldo, by the time going back to 1980s, he conducted a large cohort study called MAX, Multicenter AIDS Cohort Study. There, the subjects were all male, men having sex with men. That's the population. And none of them at that point were HIV positive. And the study was conducted at 4 different locations: Pittsburgh, Baltimore, Chicago, and Los Angeles. And a variety of biospecimens were collected from these individuals. And they had the foresight, and as he keeps admitting, that they were lucky to have collected stool samples back then. I mean, they were collecting everything that they could get. Because at the time, nothing was known about the disease, HIV, and they wanted to explore it. They even collected, I think, toenails and everything of that kind. So we had the access to all those specimens. And in addition, they obviously collected the usual characteristics of the subjects like age, race, and socioeconomics and all kinds of. So that was the type of data we had, I had access to. So he said that, well, we can explore these data. So just to give you a brief understanding of the data. So to begin with, there were 265 men who started in the beginning of the study. And of them, 156 never seroconverted. They stayed HIV negative. And 109 seroconverted in about 6 months or so. This is a rough approximate time. Among those who seroconverted, about 32 of them developed AIDS within 5 years. In fact, among these 32, about 5 to 10 actually developed AIDS within the first 2 years. But the sample size is too small to narrow it down that way. But we have time to event data, so we can do more detailed analysis in the future, which I am not describing now. And another 31 or so people developed AIDS between 5 to 10 years of seroconversion. And then there are a group of 46 people who either never developed AIDS or they developed AIDS much later, 10 years or more. So clearly there's a lot of heterogeneity among these subjects, different times at which they're developing AIDS or even seroconverting. So it goes back to our question, why there is so much of heterogeneity? Can we try to understand that? That was the question we tried to answer. So we wanted to understand at the baseline. So we want to understand, to begin with, what are the changes? Because once the infection comes in, then a lot of changes take place. But before the developing infection, are there differences among these people? That's the question we're trying to answer. So throughout this talk, I'm referring to biospecimens collected at the first visit. I'm not talking about after seroconversion. Only when I'm talking about age, then I'll talk about the second part. But all the HIV results I'm going to present today is based on the biospecimens at the first visit. So we did the usual things. We looked at the CD4, CD8 ratios of seroconverters and non-converters at the first visit. Throughout this talk, NC would mean that these are the non-converters, and SC would denote HIV positive or seroconverters. And as you can see, there was really not a statistical difference between the two groups of men at the first visit. However, when you go to the second visit, so this is the only time when I'm showing you the second visit data, clearly there was a shift. There's a reduction in the CD4 count, and that's what you would expect those who seroconverted. Now, we know that microbiome plays an important role in inflammation and immune response. There are a lot of studies along those lines. So therefore, we went towards microbiome. We wanted to understand if there are differences in the microbiome among people. And again, these are very old samples. We are looking at specimens collected in the 1980s, so it's about 40 years old. So we didn't want to be too brave and go look for the shotgun metagenomics type of analysis because that costs a lot of money. So we wanted to just do 16S to see if we can even understand the microbes that are differentially abundant at that level. So we focused on the 16S rRNA, which is highly conserved among bacteria. And we focused on the V3, V4 regions of the gene. So we now looked at when you get a microbiome data, the first set of analysis we typically do is diversity analysis. We want to see if there is differences, if there are differences between groups in terms of the within sample diversity. So alpha diversity is one measure which measures within a sample, we have a big spread of different types of bacteria. Okay, and that's measured in multiple ways. There are different measures of alpha diversity. One is people look at species richness. So in other words, you want to see how different types of bacteria you have. The other one people look at is the Shannon's entropy, which is what I'm describing here. And Shannon's entropy basically looks at the diversity. It quantifies the diversity by taking, it's almost like a variance, but you should not use the word variance for this type of data because these are compositional data. So what you're seeing here is that there is really no difference between the two groups of people in terms of the diversity at the first visit. That surprised us because usually you start seeing some differences in the diversity. But diversity is a misleading concept, and although we all use it, because here is an example, of consider two subjects, and the right panel subject one. What you're seeing is there are 4 different microbes here. This is just a toy example, A, B, C, and D. And both groups have both types of taxa, A, B, C, and D. But and also, if you look at the distribution, 60% on the left-hand side, the distribution looks like 60%, 10%, 10%, and 20%. And the right-hand side, again, you're seeing the distribution of 60%, 10%, 10%, and 20%. So any measure of diversity you use would conclude that both these subjects have similar alpha diversity. However, what you're seeing really is these two subjects have very different composition. The subject A has a lot more of the blue variety compared to subject B, whereas subject 2 rather, and subject 2 has a lot more of the gray variety compared to subject 1. So what you're seeing is that the compositions of the microbes can be different, even though the alpha diversity can be same. And this is what we are trying to understand now. We are trying to understand the differences in the composition or differences in the abundances of different taxa between the groups. So we want to test hypotheses such as, is the blue variety similarly abundant between the two groups? Gray variety, is it similarly abundant between the two groups, and so on? So that's the kind of hypothesis we want to test. So using the microbiome data that we collected, we, this is a 16S data again. So we compared this seroconverted group with the non-converted group. And I like to orient you to this figure. On the x-axis are the different taxa. The letters are the genus, and the words are the different species within the genus. And the y-axis, what you're seeing, it represents the log fold change in the abundance. So you're looking at relative to the non-converters, the seroconverters, the abundance of each of the taxa. And what you have is the estimates in each bar is a fold change in the log scale. And you're also seeing 95% confidence interval around each of them. And the stars typically describe the level of significance we have, p-values less than such and such, after controlling for multiple systems. And these results are very striking, and I'll go through them slowly. First, you see a large increased abundance of Pravitella stercoria among people who seroconverted versus those who did not. And in fact, it's a log to the base 2 scale, so it's roughly about 3 times or so is the increase in the abundance for those who seroconverted versus those who did not. And if you look at the ones, these are circled here, the Bacteroides here, these are all the species of Bacteroides. And they all are reduced in abundance among the seroconverters versus non-converters. And similarly, you see 2 species of Alistripes, which are also reduced in abundance among the seroconverters compared to non-converters. And Ruminococcus is another important bug that's also reduced in the abundance among the seroconverters. And then you have Akkermansia muciniphila, which is also reduced in abundance. I would like to walk through each of these bugs slowly because these are very interesting and important bugs which actually explain what is going on in the disease progression. So I'd like to start with, all these bugs in orange are all really commensal bugs which are in the gut for the maintenance of the gut and gut health. So if you look at Akkermansia muciniphila, this has an important role in mucin degradation. Basically, it's cleaning up the gut, so to speak, and in the process, it produces acetate and propionate, and which serve as the substrate for other bacteria to grow and as well also for the host. And it actually helps butyrate producing bacteria to grow in the process. And it's also involved in tissue repair in the intestinal mucosa. And it plays an important role in the integrity of the intestine, essentially. And it's anti-inflammatory, and it regulates body weight, and it's also involved in immunomodulation. So this bug has a very important role. By the way, this bug is in the phylum Verchomicrobia, which is not very abundant in general, but this particular taxon is extremely important for the gut health. So this is reduced in the abundance among those who developed, who seroconverted. And there's a lot of literature on it. I'm just flashing some interesting review paper. And if you look at the various species of Bacteroides, Bacteroides fragilis is an important bug again. It's a commensal bacterium. Among its roles, it prevents the expansion of Th17 cells. This is important because as these progress in response to infection, there's a potential damage to the mucosal barrier. So this is very tightly, the expansion is very tightly regulated, and Bacteroides fragilis plays an important role in that. It's directly involved in immunity, gut immunity, and also produces neurotransmitter GABA. And some of the HIV patients at the beginning stages, they experience diarrhea. And this GABA transmitter is involved in the motility in the gut and potentially is related to the events you're seeing in the HIV patients. And next we saw Bacteroides ovatus. Again, this is an important commensal bacterium. It's anti-inflammatory, and there is a development of probiotics based on this bacterium, as it produces again acetic acid, propionic acid, and isobutyrate and isovalerate acids. So it's involved in the short chain fatty acid production. Again, there's a lot of literature on these bugs. Next, I would like to describe to you the Bacteroides member that we found, which is uniformis. This is again an important gut bacterium. Again, this is involved in the production of IL-10. This, together with fragilis that I just mentioned, they both are involved in the gut health, not to expand the Th17 cells. And it's also involved in metabolic and immune dysfunction. Again, there's a lot of literature on these bugs. The reason I'm bringing up all this is because these bugs that we identified are important commensal bugs which are lost or reduced in abundance for the people who developed HIV infection. To summarize what we just got, so in the context of short chain fatty acids, several of the bugs that we identified, which are highlighted in yellow, they're all involved in the production of acetic acid or acetate. And acetate has a lot of functions, including nourishing the new butyrate producing bacteria and protection against pathogens. So these are reduced among the people with HIV infection. And then we identified several bugs in this study which are involved in the production of propionic acid or propionate. And again, this is involved in the regulation of appetite, but more importantly, anti-inflammatory. And then lastly, we identified several bugs which are involved in the production of butyric acid, Faecalibacterium and Euclidibacterium, which I did not talk much about, but they're involved. And again, this is important in prevention of leaky gut, and that's critical for this study. So in summary, the bugs we identified are involved in this process that I highlighted here. End of the day, leaky gut becomes a phenomenon that's of importance here. Why? This is where the Prevotella comes into play. We saw an overabundance of Prevotella among these people, especially Prevotella stercorea. What happens is that because of leaky gut, the Prevotella enters the tissue and starts the cascade of events for production of various IL-1, beta, IL-6, and so on, and in the production of Th1 and Th17. This becomes important now because the HIV infection binds to the CD4 protein on the surface of these Th cells. And now if you have a co-receptor, which is CCR5, then because of CCR5 presence, the CD4, the HIV infection gets into the cell. And now that's how it starts propagating. So some people have, because of some genetic mutation, don't have the CCR5 active. In those cases, even if they're exposed to HIV infection, they would not have the infection. And so now in the process, the increased number of CCR5 positive Th cells will basically spread the transmission. And again, this bug is involved in, therefore, the mucosal inflammation. It's a pro-inflammatory bacteria. And again, there's a lot of literature. So in view of all this, we went back and looked at cytokines, how the cytokines look. And sure enough, as we expected, the IL-6, LBP, and CD14 and CD163, they're all elevated in people who later developed HIV infection. So there's a sequence of events taking place. Then a natural question to ask, we did at the family level now, because Prevotella species happens to be highly abundant among people who are HIV positive, and many of the Bacteroides species are reduced in abundance. The question is, can the ratio of the two predict HIV development? So if you look at the ratio of these two at the family level, because we went to the family level because at the genus level, the sample sizes were not very large. So we pooled it up to the family level. And when you look at that, sure enough, the ratio of these two in the log scale are differential between the two groups, non-converters and seroconverters. In the seroconverters, the ratio is elevated compared to the non-converters. And if you were to do a simple logistic regression, you'll find that the probability of seroconversion increases with the ratio. In other words, people with higher values, higher levels of this ratio are more likely to have seroconversion. The next, we try to look at the short chain fatty acids, whether the propionic acid correlates with the CD4, CD8 ratio. And sure enough, propionic acid is correlated positively. So increased levels of propionic acid is protective. In other words, you have increased levels of CD4 counts, that is good. The other three short chain fatty acids are not. And part of the reason perhaps is because of the couple of outliers which we did not remove, we left them there. And if you did not have those outliers, perhaps acetic acid might have been also positively correlated. But bottom line is the short chain fatty acids are going in the same direction as what we would expect. So next we looked at time to AIDS. So once you are seroconverted, are there differences among people who are developing AIDS sooner than later? So we use the delayed converters, those who took more than 10 years to develop AIDS as the reference group, and we are trying to compare the other two groups. Again, here we are now looking at the data after seroconversion. So these are now the biospecimens used at this time point, because we wanted to start it. Once you're seroconverted, what are the chances of developing AIDS? And when we do the analysis again, we find a similar story. Again, Prevotella turns out to be a major player among those who could seroconvert sooner than much later. It's highly abundant among them. And the Ruminococcus family, like Faecalibacterium, is kind of reduced in these people who are converting sooner. And again, we looked at the people who developed AIDS between 5 to 10 years against those who took a longer time. And once again, you see some common names, which are, so here the surprising thing is the Lachnospiraceae has got some members which are, so the big fat red you're seeing, there are 3 of these members which are increased in abundance among people who are developing AIDS sooner than later. But Blottia is one member that is reduced. So what this suggests is that, and doing analysis at a very high level is not always good. We may want to go to the strain level if we can. And unfortunately, we didn't develop metagenomics analysis here to investigate at that lower level. So in summary, if we were to compare our findings, which is before seroconversion, and the literature, which is often after seroconversion. And what we find is that before seroconversion, there is intestinal dysbiosis characterized by the changes in Prevotella and these various other commensal bacteria. And that's discovered after seroconversion as well. Often people think that once you're seroconverted, you have got dysbiosis and things change. That does happen. But what we are talking about is before the infection, these changes are taking place. And again, they reconfirm what we discovered in our analysis before the infection. Again, as I just said, the ratio of Prevotella to Bacteroides is very predictive of HIV infection. and propionic acid positively correlates. And if we had a better or larger data, perhaps we would have seen other short chain fatty acids also collaborate, especially butyric acid might have been associated. And the cytokines that are seen to be elevated in our data, again, literature has shown that these things are elevated after seroconversion. So we conclude by developing a plausible model. So first, we believe that there's a gut dysbiosis for whatever reason. This leads to leaky gut and translocation of the bacteria, especially Prevotella and others. And this leads to local systemic immune activation. And now if there's a HIV infection lurking around, then they latch on to these the TH cells, and now you start expansion going to HIV conversion or seroconversion. And that leads to AIDS infection. And now the question obviously is that what causes this this dysbiosis? Why these SC group of men have different microbial composition prior? And that's the story we are trying to build now. We are looking at various sexual activities and exposures and so on, and how they correlate with these this whole chain. In our future study, we have a proposal out there where we are trying to look at the oral microbiome along with this gut microbiome, and we are looking at the also the metagenomics to get to the functional aspects of these microbes and go to the lower level of the taxonomy. And with that, I'd like to thank my student, Huang Li. He's a postdoc currently, and my collaborator, Dr. Ronaldo at University of Pittsburgh, and Dr. Chen at University of Pittsburgh. Both of them played a very big role in educating me about HIV and AIDS, because I knew nothing about it when I started. And in the process, of course, hopefully they learned something about microbiome analysis from me. So thank you so much.
So Dr. Padada's talk is open for questions. Just raise your hand. I guess I'll ask the first one. So you can manipulate the gut microbiome using probiotics or antibiotics or things like that. So the question would be, can you manipulate the microbiome as an intervention and reduce the likelihood of seroconversion?
Yeah, that's a good question. So there are studies out there which are going in the direction of probiotics and so on. I did not mention about antibiotics in this whole talk. That's the elephant in the room. I mean, we are looking at the antibiotic because many of these men at the beginning, because this was like, as my colleague says, it's Wild West. At the time, they did not know what treatment to be used. So people falling sick, and some of them were given antibiotics. And in fact, they did bad to the system than good. So we are teasing out that part of the data which I have not described at all here. And the probiotics is a direction which is of interest, not to us, but a lot of people are interested in it. Again, identifying these commensal bacteria, especially Bacteroides families, there are several of the targets coming from there.
Irma?
So are these particular taxa, the Prevotella, do you see this kind of a pattern in, are there other diseases that are characterized by similar trends in those two?
Yeah, I do. So in fact, Prevotella is a very crazy object. And that's why I think we need to go down the taxonomy, because Prevotella stercoria has this crazy behavior, and there are other members. But if you look at Prevotella copri, which is actually seen to be important for women who during pregnancy to have it, because the children born have certain resistances to certain types of allergies and so on. So there is that literature coming. So it's hard to, this is, I think the way I describe this, it's at a very high level, so to speak, and we need to really drill it down. So to answer your question, Prevotella has been seen, in fact, obesity and so on, the Prevotella ratio, again, Prevotella to Bacteroides ratio plays a big role in, because they're all related inflammation. So there is that.
Any other questions? Trevor?
Thank you for your talk. You got my attention very early on when you said that traditional statistical methods. Sorry, thank you for your talk. Thank you. I get that. So thank you for your talk. You got my attention early on when you said that traditional statistical methods cannot be used for compositional analysis. And but I don't think you really commented on the statistical methods you then used. And I'm actually thinking about the exposome, which is another topic we're dealing with during this council meeting. And, you know, the exposome is dealing with chemical mixtures. So maybe you could talk a little bit more about statistics for compositional analysis.
Thank you so much. I mean, that's a loaded question. Because compositions come about in a lot of data we see actually, but okay, when we started way back with the microbiome, it was very difficult to convince even statisticians that these are really compositional data. And eventually they agreed and they started, now it has become the thing, everybody uses compositional methods. And that can apply to some of the other omics data as well. It depends upon the technology, how the data are being developed and generated and processed and how you can interpret them. If you view the data as relative quantities, so for example, if you think of single cell data, the data that you generate really are total counts in some sense, and you got relative quantities of this. So in some ways, you can think of single cell data to be also compositional. And it's a topic that people talk about, but they move on. So to answer your question, the mixture data, again, if you think of if I'm exposed to a bunch of chemicals, I'm exposed to certain parts of this chemical and certain parts of that chemical and so on. If you think that way, then it's a it's a compositional data then it depends on the scientific question you want to ask from that point on. And if you're trying to make comparisons about, does this composition differ from that composition, some phenotype? So I'm developing some disease. Does 10 parts of this chemical and 20 parts of that and 30 parts of this chemical, is that bad compared to 30 parts of this and 20 parts and so on? Then you're talking about really as a compositional data. Then you have to think of analysis along those lines. And I think all fields are now picking up on as compositional data now for a lot of different applications. So again, my answer to that question is I have to be cautious. I mean, look at the question I want to answer and what is the data that I'm really seeing and how do I want to interpret the data.
So do you think that one of the approaches is hierarchical clustering?
Hierarchical clustering is a common approach for, again, depends upon the point of clustering is to group things together of a similar kind for a certain phenotype. Their compositional methods may work. So let me step back a bit again. If you are trying to do some predictions, okay, if you're trying to predict something, ignoring compositionality is forgiving because at the end of the day, you want to predict some linear combination affecting some outcome. But if you are trying to make comparisons between groups, just like I was trying to do, then you have to acknowledge the compositionality. I think it comes to the formulation of the question.
Thank you.
Okay, I'm not seeing any other questions, so let's thank Dr.
I think
Padada.
Look to your right.
You're not using your hand, Rick.
Oh, sorry.
Rick.
Actually, that was a great talk. Very, very interesting.
Thank you.
So the composition of the microbiome is always it's something that I've been very interested in. And I think there's some data out there to indicate that the genetic background of an individual can also influence the composition of the microbiome. Is that correct? That's correct. And I have another question.
That's right.
So, okay, if that's the case, then what's the possibility that the driving force for a seroconversion is not the microbiome, but the microbiome is simply a reflection of some genetic combination that is actually the driver
for seroconversion.
Absolutely. And that's a difficult thing to parse out, but you're right. I mean, that's the host and microbiome interactions play a very big role in everything. So we are simplifying the problem substantially when we are doing what we are doing. But end of the day, it's a puzzle. I mean, you're trying to discover all pieces of the puzzle and then make a story in the end. But so that's a big leap that we are making, which we are not discussing at all. And a lot of literature is emerging along those lines, what you just said.
Just another quick question. Were there any questions about the diet of the individuals? I think one of the things that clearly comes through is that if you eat your vegetables, you have plenty of complex carbohydrates that somehow nurtures a certain type of microbiome. So could it be that diet is also a variable here?
It is. I think, as I said, the precursor to this gut dysbiosis is a bunch of exposures. And as the follow-up study that we are doing right now, we are taking into account a lot of that. But I have to say, back in the day, the data that were collected, the diet data are not very rich in our study. So we'll have to just take what we have. But these days, there are a lot of refined studies going on. In fact, the other thing I forgot to mention is that in this study, none of the subjects were exposed to any of the modern treatments for HIV and AIDS. So this is really back in the day. And there could be a lot of other confounders that could be going on, which we are trying to tease out. But it's a first pass at the problem, I think.
Okay, no other questions. Let's thank Shamal again for a wonderful talk.
Thank you.
Okay, our final presentation of today's open session is the NAEHS Executive Officer, J. Ingrid Mathis. Jay.
Thank you. And I'm glad to be here today. I thought I'd talk with you all a little bit about my role as the Associate Director of Management and the Executive Officer. And specifically, I'll focus on my background and experience because I haven't had a chance to meet all of you. I know I've met some of you. So I'll talk a little bit about my background. I'll also talk about what it means to be the Associate Director for Management and give you a little sense of the Office of Management as an organization, as well as our vision and priorities going forward. So in terms of my background, I've been doing management and leadership of administrative operations for about the past 25 years or so, and primarily in a research setting. Most recently here at NAEHS, I was in the intramural program before stepping into this role. And prior to that was actually over at the EPA across the lake with my colleague, Andrew. It was nice seeing him today. And then prior to that at the UNC Injury Prevention Research Center as a grantee. So very much aware of what it means to be on the other side of the fence and soliciting funding through grants. And then I also previously had some experience at HHS and the Administration for Children and Families. So I have a background in social work and psychology, which comes in handy a lot in a role like this. And speaking of the role, let me just tell you a little bit about what is included in the oversight as the executive officer. It's a hefty lift. At the center is really about supporting the science, right? That's why we're here in the Office of Management. It's to make sure that we position NAEHS to advance the science. Key to that is our workforce. And oversight of, we do have a union here. So part of my role is making sure we adhere to our collective bargaining agreement, making sure that we've got strategies in place to support recruitment of staff, retaining our staff. So that's a big part of what I do. You all heard Rick talk about our budget. That sits within my purview as well. My office is responsible for developing the budget, executing that budget, tracking utilization of funds. We are the NAEHS, so sustainability is a huge component of what we do, and I'll talk a little bit more about that in a minute. Contracts also sits under the EO's realm, and with that, contracts in probably every single area you can imagine, from our security and safety. Many of you experienced our armed guards coming into the community day on campus, supplies and logistics to make sure we've got the work products that we need in the labs and in our extramural program as well to do the jobs that we do. Space and facilities. So the renovation of this space, which many of you may remember, didn't look like this before the pandemic. And even throughout the Institute, there have been other upgrades that have been made. So that also falls within the Office of Management. Our Child Care Center, which we share actually with the EPA. We have a shared contract as we do with other initiatives. And this is an important one for especially our population of folks who need to have their children close by. We've got a clinical center under Dr. Hall's leadership, and even before I stood into this role, I think the relationship between the Office of Management and the clinical center was strong, especially when we were in need of an asymptomatic testing site during the height of the COVID pandemic. So things like that are critical to having the kind of relationships and collaboration I'll talk about in a little bit. Accessibility needs. I'll mention we have a great library as well. Travel, so to support travel to meetings like this, and the bureaucracy that comes with travel to meetings like this, and as well as coordinating with federal and state and Durham County governments, since we do have a pretty strong emergency management program, and that requires us to maintain close communication with Durham. So that's a lot. It's a hefty lift. And the nice thing is I don't do it alone. I've got a great team of colleagues that I work with who support me in every single area I just talked about. And you'll see them all represented here. To give you a sense about who we are as an organization in the Office of Management, I should mention our campus is about 1,700 people strong. So 1,700 people, whether as federal employees, contractors, postdocs, postbacks, our trainee population, guest researchers, special volunteers. It's a diverse mix of folks. And we're charged with providing the services I just mentioned here. I'll come back to budget formulation because that's obviously critical to us working with the extramural community and getting grants out the door. I already talked about our acquisitions folks who are key to all of the day-to-day that goes on and procurement of services and supplies. Ethics is an underpinning for everything we do. As many of you are aware, if we have senior officials or even just to abide by the basic rules and regulations that we're all required to abide by in the federal sector, it's important to have a strong program, which we do. From a campus and security campus operations and security standpoint, I'll mention that this is a large campus. Those of you who have had the time to walk it can see the breadth of it. It's actually 500 acres, 509 acres. We've got over 1 million gross square feet of space for our labs and support systems, support services here. So it's a hefty lift to make sure that, especially in a space where we've got aging infrastructure, that we've got our top-notch staff who's on top of things to help us continue to do the work that we do. And I'll say we've got partners at NIH in our Office of Research Facilities who are phenomenal in helping us to maintain the campus. We've got a very strong health and safety program and environmental sustainability program. There are actually 2 certifications you all are probably aware that we already have here that happened well before my tenure into this role. One is that we're a net zero energy facility, which is fantastic, which means that we consume no more energy than we are able to generate. And we do that in a variety of ways here. We also have most recently, just around the start of the pandemic, actually earned an international certification known as the Well Health Safety Certification. We're the only federal agency that has it. And it basically is a testament to both our air and water quality management here, the on-site services we have for health resources, such as the flu vaccine clinic, COVID vaccine, when we were able to do that, our cleaning and sanitization processes. So it's kind of a big deal. We're proud of that. I've already talked about travel and just administrative facilitation and coordination in general. I mean, things like this hybrid meeting, we work closely with our other partners here at NAEHS, including our communications office, our IT folks, to make sure that we get better at doing these sort of hybrid meetings. In terms of our footprint, we're about 10% of the overall NAEHS population, and almost evenly split with feds and contractors. And our budget is roughly 3% of the overall NAEHS budget at $3.1 million. So just to kind of frame things more specifically in terms of how we support the grant making process, our office, our financial management branch, about 40% of our staff support the grants program. And they do that in a number of ways. First of all, with RFA process, they work really closely with our extramural colleagues to put in appropriate funding for the grants budget. Once decisions are made about which grants are funded, we certify availability for those funds. Do a lot with tracking to make sure that the funds are spent appropriately in the appropriate sub-mechanism. And also just to make sure that things are flowing. You know, you never want to get to a space where you're blocking the use of these funds. So we work really hard to make sure we do this in partnership. So I'm going to pivot a little bit. I talked with you about who we are. Now I want to just talk a little bit about our vision and how we do what we do. Our vision, as I mentioned before, it's really to provide infrastructure and administrative support services for the Institute and to advance science. And I really see as key to that these three bolded areas of innovation, collaboration, and inclusion, which I'll talk about in just a few minutes. But what I've done is put in place, since I've started in this role, which hasn't been very long, I've been here as CEO since July of 22, and quickly put in place this framework that I refer to as WWP for workforce, workplace, and processes. And it really is kind of the governing structure we use in making decisions about how we approach the work that we do on behalf of the Institute. And from a workforce standpoint, it's really focusing on strategic recruitment, especially knowing that we're evolving as an institute and we're getting into things like climate change, making sure that we've got strategies in place to support us recruiting the talent that we need. And importantly, supporting the talent that we've got here on site as well. I mean, we know that in the space where people can move now, there's a lot more remote work opportunity. We want to retain our staff, and figuring out how to do that from a standpoint of career development and advancement is key. And so part of what we'll be doing or are doing is really looking more broadly across the Institute to position us to have these institute-wide plans for recruitment and retention and succession planning. This middle box is workplace, and you'll see the first bullet there is really on enhancing a hybrid work culture. I mean, this is our norm now, having these hybrid meetings where we have some folks on site, many folks who are working remotely. I'll tell you that about 10% of our workforce are remote workers right now, and almost 70% of us telework to some degree. And so it just requires us to get really more, experience in having successful meetings like this, where folks who are on site and folks who are on the Zoom have parity in terms of how they can interact. And we're working hard to try to make that even more seamless for everyone. I've talked about or noted here the development of physical and virtual infrastructure. We've got an aging campus, an aging building. We're hopeful that in the next couple of years, we'll see the construction of what we call our CCSB, which is our Clinical and Computational Sciences building, and it'll bring us all together. It'll be an addition here onto this building, and will eliminate eventually the need for an off-site building over what we refer to as the Keystone Complex. And then I also want to, as we talk about workplace, I really like to reiterate the importance of collaboration, which I'll talk about in just a few minutes, as well as just promoting this climate of workplace civility. We've talked a lot, or you've heard some today, about diversity, equity, inclusion, and accessibility, that DEIA. And in a space where we're talking more publicly and socially around representing different viewpoints, encouraging different viewpoints, that also comes with just having basic civility and making sure that wherever you are in the spectrum of an employee, whether you're a contractor or a trainee, or whether you're a veteran, or wherever you are, that we have this culture where folks feel that there's a basic level of civility. And then finally, our processes. One of my areas of focus is on streamlining the administrative bureaucracy. The last thing we want to do is, we work for the government, obviously. There are going to be rules and requirements, but there are things that we don't have to do that we're doing. There are things that I'd love for us to just strip away and just do the bare minimum, so that we're, again, focused more on the science and less so on doing things just for the sake of doing them. So importantly, and you heard Rick talk earlier today about the strategic plan. we're very clearly aligned with that. We want to make sure that as the plan evolves, as the direction of the Institute grows and evolves, that we're agile and we're able to evolve with it. And part of that is focusing on the leadership values that I'm guessing you all may have heard about at some point. As the senior leadership here has stood up these 5 values of workforce, innovation, communication, collaboration, and distributed leadership. It actually all fits in really nicely with my emphasis on inclusion and innovation and collaboration, especially when it comes to this final bullet here on the DEIA and C. I like to add the C for civility. But we've got a lot of work going on in the space where almost from a grassroots effort, which is really nice to see that employees are really engaged around diversity and equity and inclusion, accessibility and civility, and really coming up with ideas of things that we can do internally. So that's Rick referenced this earlier. It's really a focus of our senior leadership, and we're starting to look more at how we can meet in the middle, both from the top down and bottom up, and implement programs that will be specifically geared towards DEIAC. And I'll talk a little bit about that. So again, keeping in mind my emphasis on innovation and collaboration and inclusion, a hybrid work environment is here. We're obviously needing to continue to focus our efforts there. We have set up more recently a hoteling effort. So for those 10% of remote workers that we have who have occasion to come on site, there's now software available for them to go on site, reserve some space, and work as they would if they just had their own office or cubicle here. We're also focusing on onboarding. One of the things I'm keenly aware of is we've recruited a lot of new talent who started during the pandemic, some of whom never set foot on the campus other than to pick up their badge. And you can imagine how difficult it is to get integrated into a community where you just in some ways feel isolated. So we're rolling out a pilot program in Office of Management that will eventually extend all up to the NAEHS. That's a buddy program to help link newcomers with more seasoned staff and give them a regular point of contact throughout their time here as they get acclimated. We're also looking into enhancing our offboarding efforts. So no one likes to lose staff, but it happens. And we're recognizing that that's been an untapped resource. I mean, we have had exit interviews in place, but don't really encourage or haven't really seen enough people use them. And so we're placing more emphasis on getting feedback from people. And yes, it's as they leave, and the majority of people who leave tend to do so due to retirement, but there are also a number of people who leave for other reasons, and knowing what those reasons are so that we can do some work on the back end to mitigate further loss is really an area of focus for me. And I talked about enhancing our physical infrastructure. I'll just mention too, I talked about being agile, and in the time where COVID was at its highest, one of the things that Office of Management did with our IT shop and our communications group, again, was set up an outdoor meeting space. And so as things change like that, we're continually looking for opportunities to innovate and welcome those, because I think it helps position us for change and gets us more acclimated to doing those sorts of things in the future. Let me just talk briefly about collaboration. I mentioned, or did I mention? I meant to, that we've got a union. We do have a union environment here. Roughly 20% of our employees are in the bargaining unit, which does create somewhat of a, this push and pull where you're trying to accommodate and adhere to the collective bargaining agreement while not creating any sort of disparity, because there's differences and nuances in treatment sometimes. And one of the things I'm excited about is reigniting what's called our Labor Management Council. It existed for a long time. It kind of fell dormant during the pandemic. We're reigniting that. And the intent is really to start conversations early in the process and approach them more as partners so that we can reduce some of the adversarial nature that tends to come when you've got a labor management environment. We're also heavily collaborating with NIH in many ways through our Office of Equity, Diversity, and Inclusion, especially as we implement some of our DEIA activities. Especially with the Office of Human Resources, we're always looking for ways to streamline, especially recruitment. I mean, Rick, hearing you talk about the ARPA-H, I'm jealous that they can move as quickly as they can to hire people. And I'd love to get us to a space where we can do some of that as well. And obviously our Office of Research Facilities, which I mentioned in terms of helping us with our campus needs. I've kind of hit on inclusion. I will tell you that one of the things, again, we're doing, and this was also an effort that was generated from the bottom up, is establishing a formal mentoring program. I mean, mentoring occurs in many pockets throughout the Institute with our trainees, for instance, and even within some of our divisions. But we want to formalize that and give everyone an opportunity to have a mentor, especially those folks who are just looking to advance in their career. And advancement may not mean promotion. We can't promote everyone, but we can certainly position them wherever they're headed on their next journey. And hopefully it's still within the realm of public service. We can help get them to a space where they're feeling like they have what they need to be successful. And I'll just highlight too, I've been privileged to sit on a committee with our NIH Office of the Director on awards recognition. And most recently, that group was responsible for initiating guidance that we hope will open up opportunities to include even more of our staff from all ranks. I mean, I think what we were noticing and what the Office of the Director was noticing was that there tend to be the same numbers of people recognized or the same types of positions recognized. And so our intent is to open that up a lot more broadly. And again, this is one effort to be more inclusive and encourage folks at all levels of the organization to be nominated and recognized for their value. So in summary, the WWP is alive and well. We're going to continue to focus on our workforce, our workplace, and our processes, and continue to be guided by those 3 core values I mentioned of inclusion, innovation, and collaboration, as well as the NAEHS core values. And we're excited. I mean, there are many partnerships available to us that we recognize as thought leaders here. We feel like we can influence what happens at NIH. And likewise, learn from NIH. I mean, we're unique in that. In my role, my counterparts at our sister ICs don't have the same scope of responsibility that I have. So they don't have to manage their own campus. They don't have to manage their own childcare center, for instance. They don't have to worry about a security force or renegotiating contracts for that sort of thing. So in many ways, we're leading NIH and want to continue to do that. And I think we're well positioned to do that going forward. And I think that is it. I'm happy to take any questions if folks have any. Rick.
Can I just make a quick comment to maybe start the Q&A? Jay, first of all, I want to thank you for taking the opportunity to describe what it is that you do. And I think it should be, and we thought it was very important that council hear this, because all of the things we talk about at council, whether it's getting the grants out the door, getting your travel reimbursement, making the campus energy efficient. I mean, all of the things are really attributable to Jay's capable leadership and the engagement of her staff in the Office of Management. So I just want to personally thank you. I suspect I'm speaking for all members of council and senior leadership. So again, thank you for the presentation. And actually, if there are other comments or questions.
Thanks, Rick. Yes.
Yeah, I just want to say that I didn't realize that this was a net zero operation, which is pretty impressive. I mean, we've been working really hard to try and get our university campuses on board with just, an aspirational, 2030 kind of situation. So congratulations on that alone, let alone all of the other. Yeah, it just struck me as this is everything. Hard to believe that that's all compacted into one person, but terrific. I mean,
yeah, thank you. Yeah, and that that happened, as I said, before I stood foot in this position. But I think it says a lot that there's that level of focus and commitment to sustain the environment. We're actually doing more in that space with solar panels, even purchasing of our fleet, our vehicle fleet we've got several electric vehicles. We're about to install some EV charging stations that our employees will be able to use if they've got personal vehicles. So there's a lot coming. There's a lot that's been done, I had a very solid foundation upon which to start. So I'm fortunate.
And maybe in one of the future council meetings, you can arrange for a visit to our new well, not new anymore, but our very energy efficient warehouse.
Absolutely. That'd be great. All right. No other questions?
Andrew.
Oh, Andrew.
Can you, Jay, you talked about, was it Well Health Certified? Can you talk a little bit more about that? I'm curious about what that
Yeah.
entailed and what that means for your campus and workforce here.
Absolutely. So it's an international certification. They have, I think, 3 or 4 different criteria. One is how well you manage your air and water quality. One is how well you provide services to your employees in terms of health services. Another is around your cleaning and sanitization protocols. So if, for example, we clean high touch areas 3 times a day here. And so it's true, we are the only federal agency right now, Andrew, that has a certification. We sit in the company, I think, of the New York Yankees and maybe JP Morgan and one of it. So it's not a well distributed certification right now, but I'm happy to talk with you more and actually connect you with a few other folks who were responsible for putting that whole initiative together, because it was a hefty lift, and it's amazing that we've been able to do that and to sustain it. And you do have to sustain it year after year. I mean, you get reviewed based on how well you continue to adhere to those specific criteria that got you the certification in the first place.
Yeah, yes, I'd like to hear more about it. I think about our campuses and how much we've tried to build for our staff and our scientists, and here 3 years into COVID, we see people opting to work at home and making the workplace that welcoming and supportive in these ways are I think some of the ways we can draw people back into the workplace.
Absolutely. In
And
fact,
all
that
the
was a
other
big
benefits.
draw when the certification was received. I mean, part of the thinking was, we wanted folks to feel comfortable returning back to work, and being able to say that, well, we're well certified, I think is helpful to many people to recognize we're taking this seriously and we care about our employees. Yeah. All
Any other
righty.
questions for Jay?
Thank you.
With that, we are done with open session for day 1.